Hi everyone, my name is Shawn Thomas and I'm excited to be here at Virtual Botany 2020. Today I'm going to be talking about the phylogenetic reconstruction of the Cornales that I did using target enrichment. This is work that I started during my undergrad and I'm continuing to work on now. So according to the latest APG, the Cornales are placed sister to the asterids with the Ericales. This is a relatively small order of flowering plants, including around 600 species, including some horticulturally important taxa like dogwoods, hydrangeas, tuberos, and others. They share some morphological characteristics like four-parted flowers or bracts, drupaceous fruits, and inferior gynoecia. But as you can see in these pictures here, they have a large morphological diversity. So when I look at the Cornales, we tend to see lots of shrubs like hydrangea bushes here. Then we also see lots of trees, such as flowering dogwood, tupelos, and the dovetail tree. But then when I look at this family Hydrostachyaceae, this looks like some kind of weird aquatic fern-looking thing. But it's not. It's actually a flowering plant. So this actually piqued my interest, given that we see lots of trees and shrubs, and then this family has these weird aquatic plants. So I want to explore it a little bit further. So looking at previous studies of the group, Fan and Zhang in 2003 produced phylogenies of the group with a few markers and got conflicting results on the placement of Hydrostachyaceae. So here you can see that the Hydrostachyaceae is placed within the Cornales, and here it's placed sister to the rest of the Cornales. Additionally, these trees, although you can't see it, they're not very well supported. Then in 2011, Zhang et al. Published another study with additional markers, and this time with better support. However, the tree was not fully resolved. Then at Botany 2019, Dr. Jenny Zhang presented a phylogeny reconstructed from whole plastid genome amino acid sequences. And here's the topology that we see here. Here we see that the Hydrostachyaceae is placed within the Cornales and is placed sister to the Hydrangeaceae and Losaceae clade. So my goals for this project were to produce a well-supported species tree using target enrichment to capture hundreds of nuclear loci in order to help solidify the placement of Hydrostachyaceae. So when I think of target enrichment, I like to use this metaphor of fishing because we're using baits to capture our target genes. Credit and thanks to Dr. Sarah Unruh for sharing this metaphor and image with me. So here's an overview of target enrichment. Looking at this first step, we see our genomic library. This is sort of like our pond with our fish, and the red indicates our target sequences that we want to capture. So in order to do that, we have a fishing rod with our baits. So we have RNA baits and these streptavidin beads. The RNA baits will bind or hook to the target genes, and then we can use a magnet to reel in our targets of interest. And then we can amplify and sequence those for phylogenomic studies. So some pros of target enrichment are that it's great at selecting target loci and avoiding repetitive elements in pair logs. However, some cons are that it requires existing genetic resources, and actual bait design is both challenging and expensive. In order to overcome the barriers to entry for target enrichment, we decided to use the Angiosperm 353 bait set. Johnson et al. Used 410 low-copy orthogroups from 31 green plant genomes from the 1KP initiative. To add loci to these gene families, they additionally included 1,100 green plant transcriptomes. They did a clustering analysis to filter to only include angiosperm sequences and selected instances where 95% of angiosperm sequences were represented with less than 30% sequence divergence. Finally, they ended up with 353 loci with 5 to 15 target sequences for each. So once we did the wet lab work, we used this pipeline for analysis. We used a program called HypePiper, which takes our trimmed reads, maps them to the target sequences, and then assembles the genes, as you can see here. So to orient you, these green boxes represent our exons, and these black bars represent the flanking intron regions. These assembled contigs are called supercontigs because they include exons and flanking intron regions. Next, we use a program called PASTA to align our sequences for each gene and use IQtree to assemble our gene trees. Then we put our gene trees into the program called Astral to get our species tree inferred from a multi-species coalescent model estimation. So for our sampling, we were able to represent all families in the Cornales and additionally had outgroup sampling in the Aracales, some asterids, and some rosids as outgroups. On average, we were able to recover 90% of genes for 158 samples. So we had some samples that we had to throw out, and we set an arbitrary 20% gene recovery cutoff. And then the remaining samples are these 158. There were 21 samples that we had that did not go through hard enrichment due to low DNA concentrations, and we removed 48 samples that had low gene recovery, ranging from 0 to 11%. Since it's difficult to look at a tree with 150 plus tips, I condensed the trees to just focus on the familial relationships in the Cornales. Tips with triangles represent a condensed node, and on this tree, branches without a value have a posterior probability of 1. So that just leaves these two branches here with lower support. So you can be pretty confident in this tree topology. So now let's take a look at bootstrap support. To orient you, this green box here represents the Cornales, and then moving out from that, this blue box represents the asterids. Sister to the asterids, we have the Caryophyllales, and then as our outgroup, we have rosids. So here we see that the bootstrap support is less than 100 at these two branches here, and everywhere else on the tree, we see 100% support. So now let's compare familial placements with my tree inferred from 353 nuclear loci versus the Botany 2019 tree, which was inferred from whole plastic genome amino acid sequences. So here we can see a majority of the clades are being preserved, such as the Cornaceae Langiaceae clades, the Nisoids, the Grubiaceae Cartesiaceae clades, and the Hydrangeaceae Losaceae and Hydrostachyaceae clade. And additionally, we're seeing that the Hydrostachyaceae in both trees is being placed in the Cornales. However, we do see some differences. Rather than being placed with the Hydrangeaceae, Losaceae, and Hydrostachyaceae clade, as we see here, we see that the Nisoids are being placed with the Cornaceae, Langiaceae, Grubiaceae, and Cartesiaceae. And additionally, we're now seeing that the Grubiaceae and Cartesiaceae are sister to the Nisoids rather than the Cornaceae and Langiaceae, as we see here. Now let's look at quartet support on a few select branches. Just to orient you, these pie charts represent the frequency that topologies are seen in the gene trees. Green represents the main topology shown here in the tree. Red is the first alternative, and blue is the second alternative. Looking at this branch here, we see about 83% support for the main topology and relatively equal frequencies for the two alternatives. So this gives us good support for this main topology. Then moving back and looking at this branch here leading to the Cornales, we see a bit lower support for the main topology, which is around 62%, and relatively equal frequencies in the two alternatives, 21% and 18%. This still gives us good support for the main topology since the two alternatives are at equal frequencies, and this is likely due to ILS. However, when we see unequal frequencies in the two alternative topologies, we can't conclude that these patterns are due to ILS. In the next few slides, let's look a bit closer at some of these branches with uneven support. So here we're looking at the branch leading to the Nisoids and the Grubiaceae Cartesiaceae clade. For our main topology, we see this being represented by 62% of gene trees with information for this branch. Looking at the two alternatives, we see unequal frequencies. We see 25% support for this first alternative, which places the Grubiaceae Cartesiaceae sister to the Cornaceae Langiaceae, and this is a grouping that we've seen in some previous phylogenies, including the Botany 2019. Then, looking at the second alternative, we see that the Grubiaceae Cartesiaceae is being placed outside of this group with the rest of the tree. Since we see this unequal frequency in our first alternative, we can't assume that this pattern is due to ILS. So perhaps there's some valid evidence for ancient hybridization or gene flow leading to this Grubiaceae Cartesiaceae clade, perhaps some gene flow with the Cornaceae and Langiaceae. Now let's look at quartet support on the branch leading to the Hydrangeaceae, Losaceae, and Hydrostachyaceae clade. For the main topology, we see 53% support, and again, see unequal frequencies in the alternatives. Looking at the first alternative, we see 10% frequency for gene trees placing Hydrange ACA and Lowe's ACA, sister to the rest of the Cornales, with the rest of the tree. For the second alternative, we see 37% frequency of the gene trees placing Hydrostachy ACA with the rest of the tree and outside of the Cornales. The majority of gene trees support the placement of Hydrostachy ACA within the Cornales, but this 37% leads me to hypothesize that there may have been ancient reticulate evolution or ancient gene flow in the Hydrostachy ACA lineage with taxa outside of the Cornales. So from these results, despite the possibility of ancient gene flow and hybridization in the Cornales, we see that the monophyly of the Cornales is well supported by the data and that Hydrostachy ACA is confidently placed in the Cornales from our support values. In the future, I want to explore some of these hypotheses of ancient reticulate evolution and hybridization using some formal tests. I was hoping to get this done for this talk, but I'm just going to blame it on COVID. And additionally, I plan to submit this work as a paper in the upcoming Angiosperm 353 special issue. Finally, I'd like to give a few acknowledgements. I started this work at the University of Georgia working with Dr. Jim Liebensmack. Shout out to Dr. Adam Buick for being an awesome postdoc mentor, and Amanda Cummings for doing all the scary wet lab work. We got most of our samples from the Zhang lab, and thanks to Dr. Lisa Picorni for providing us with some additional data. And finally, the Peers Lab, where I'm doing my PhD now. Funding for this project is supported through the NSF and the Dogwood Genome Project. And finally, thank you all for attending my talk. Please post any comments or questions in the chat box below.